Hey all you cool cats and kittens. Today we're going to go over the answer key for Friday, April 3rd. So what we're talking about are steroids and other things that are associated with the membrane, as last time we covered lipids. Big difference here, steroids are fused rings, so they're a lot more stable. Well, not stable, they're less they have less motion, I should say, and they help to modulate the melting temperature of the membrane. Think of them like planks that can push together lipids. So question one, how are steroids and lipids different? Well, here steroids have fused ring structure. They still have a polar head though, so that's how they're similar. They both have a polar head group, while lipids have a tail, a carbon tail, and steroids have a fused ring. That's how they're different. So steroids are hormones, and they're made from cholesterol based on what group they have. They'll have different functions in the body. Vitamins can also be steroid derivatives. So basically anytime you see fused rings like this means that you're from a steroid, or you are a steroid and you're from cholesterol. You can also have lipids that are from isoprenes, which have this basic unit and just repeats over and over and over again. Biochem 2 will talk a lot about coenzyme Q. It is used to move electrons around your electron transport chain, and it's made out of isoprenes. Some other vitamins are also made out of isoprene units, so when you see this repeating structure, it just kind of means that you're made out of an isoprene. And some questions you might see related to this on the test are just, do you understand the information that was presented on the previous slide? For example, is cholesterol a steroid hormone? And it's not. It's actually the precursor to your steroid hormones. What does vitamin A do in the body? It's a photoreceptor in the eye. What do glucocorticoids do in the body? They are part of metabolism signals, inflammation, and stress. And is vitamin D a steroid or an isoprene? That is an isoprene. So the membrane itself, we have a question here where we're looking at lipids. We have transverse diffusion, flip-flop, where one lipid goes from a layer to another layer, and lateral diffusion while they stay in the same layer. Speeds are very different. So why is transverse slow? So the inside of the lipid is nonpolar, while the head groups are charged and polar. So to go from one side to another, your polar head group has to go through this nonpolar body. This is very energetically unfavorable. Polar stuff likes to be around polar stuff because of hydrogen bonds and ionic bonding. So for a polar head group to go through this nonpolar membrane, it does not want to do that, and so this transition is extremely slow, taking maybe up to a day for a lipid to move from one side to another. Also, we have the image here. The lipid membrane has different fluidities. When it gets colder, it's more like a solid. Hotter, it's more like a liquid. So question 3. So this kind of ties back to our discussion on lipid properties, and now relating them to the membrane. So what are all the ways we can change the transition temperature of the lipid membrane? So we can add cholesterol, we can have longer lipid tails, and we can have less double bonds. All three of these will increase the melting temperature of the plasma membrane. And when we talk about the plasma membrane, different layers, that is the layer that's showing towards the cytoplasm and the layer that's showing toward the outside of the cell, they have different lipids, different lipid heads. So for example, macrophages recognize phosphatidylserine on the outside of the cell. That means that cell is going to die. That's because phosphatidylserine is only found in the cytoplasm for normal healthy cells. So why is this a useful marker for a cell that's dying? Because if it's only found on the cytoplasmic cell, if you ever see phosphatidylserine on the outside of a membrane, that means something really bad has happened to that cell. It's under great distress, or it's already dying and this membrane is breaking apart. So that's why it's a signal to the macrophages to come and destroy that cell. So talking about proteins, so we have different types of proteins that associate with the membrane. If you're integral, that means you are going through the membrane as shown in this image. So we have a portion that's hydrophobic and goes all the way through the membrane, and it's always associated with the membrane. We looked at these hydropathy indexes before, and basically high numbers means you're hydrophobic, negative numbers means you're hydrophilic. So you can see for this protein on the right, this part might be going in with the cytoplasm. This part, the dark green, is going through the membrane. And this could be on the outside of the cell or through the cytoplasm as well. This is how we can tell how many transmembrane domains, which is shown here, are going through the membrane. You have different types of proteins, but they have usual shapes. So here we're looking at beta barrels, so it's a beta sheet and a barrel. Here's an alpha helix that's in a barrel shape as well. And the idea is that the outside of these proteins are going to be hydrophobic, so they can interact with the lipid membrane. The inside could be hydrophilic, though, so they can allow transportation of molecules that would not normally go through the membrane. Because polar molecules really can't go through the membrane. They can't pass through those nonpolar tails. Question 4 was an in-class activity, so frowny face. Basically, we were going to look at, I was going to pass out a worksheet, and we were going to work on that together, look at some real information to determine protein is integral or not. So basically, what you should just know, since we didn't do that, be able to tell like a graph like I'm showing right here. Do you know which parts are saying that you're on the outside of the cell, or rather which parts are polar, which parts are non-polar? Do you know what a transmembrane portion of a protein would look like? What does it mean for amino acids? So that's basically what our in-class activity was going to get at. So modifications to the lipids in your plasma membrane can help link your proteins to the membrane. So you have mystric acid and palmitidic acid as two types of linkers. Here, what are these building blocks called again? These are isoprene units for different types of modifications. GPI linked proteins have lipid anchors, so they're associated to the membrane through a lipid. So this picture is actually nice because we're looking at our 3 different types of proteins. Integral, I always have to be part of the membrane. Peripheral, that means I can come off and come on. Lipid linked, a lipid is holding me there, but if I get cut from my lipid, then this protein can fly away from the membrane. Question 5 is again another one of those few questions. Is really ask yourself, did I understand what I was going through? What was going on in the lecture videos? So which of the following classes of proteins will associate with the membrane bilayer with the lowest energy and therefore most difficult to separate? Transmembrane, they go through the membrane, they cannot be removed from the membrane, they're always a part of it. So that's why it's C. And which of the following is not found as covalently attached anchors in lipid-linked proteins? Cholesterol and sterols. So sterols can be hormones. Cholesterol really just affects the fluidity of your membrane, how liquid it is, how solid it is. So you're not going to get protein stuck to cholesterol. That's not really its job. With that, ends this video. If you have any questions, as always, please don't hesitate to email me, and we can work those out together. Otherwise, I'll see you in the next video.